This topic is photosynthesis. Photosynthesis, the name itself says that the food is produced by the plant in presence of sunlight. The green plants and all the chlorophyll-bearing bacteria like cyanobacteria, algae are autotrophic and synthesize organic compounds by using simple inorganic compounds in presence of sunlight. So therefore, all heterotrophic organisms are dependent on food produced by or prepared by autotrophs for survival. All life forms on Earth are dependent on sunlight for energy directly or indirectly. Thus, the use of energy from sunlight by the plants to do photosynthesis is the basis of life. There are various experiments conducted related to photosynthesis to show the presence of starch as well as um, the main ingredient essential for photosynthesis is carbon dioxide. So green plants which are exposed to sunlight show the presence of starch and also certain experiments are conducted to show the carbon dioxide is essential for photosynthesis. So basically, photosynthesis is an physiochemical process, also called as anabolic process, by which green plants use light energy to synthesize organic compounds. Photosynthesis is important for two reasons. It's primarily the source of food for all living organisms. It is also the only process responsible for release of oxygen in the atmosphere by green plants. Various experiments are also conducted to show the um, role of air in the growth of plants. Next, we'll discuss the organelles which help photosynthesis. The site of photosynthesis in plants are the green leaves and to some extent in other green parts of the plants, and it takes place in cell organelles called chloroplast. So here you can see the diagram of chloroplast, which are arranged in form of a stack of points, and they are found in large numbers in the mesophyll cells of the leaves. Chloroplasts align themselves along the walls of the mesophyll cells in such a way that they get maximum quantity of incident light. Now they will align with their flat surfaces parallel to the wall under low or optimum light intensities and perpendicular to the walls when the intensity goes very high. So it's a clear division of labor within the chloroplast. The membrane system, which is responsible for the photochemical phase, where synthesis of ATP, adenosine triphosphate, and NADPH occurs, whereas the stroma has enzymes for the reduction of carbon dioxide into carbohydrate, which is also called as starch, and formation of sugars. The pigments are distributed in the membrane system. Next is the pigments involved in photosynthesis. The primary pigment involved in photosynthesis are chlorophyll A and B, along with carotene and xanthophyll, which is also called as carotenoids. Here you can see the chemical formula on the structure of chlorophyll A, B, and xanthophylls. Now, the chief and main pigment associated with photosynthesis is chlorophyll A, as I said earlier, and the other accessory pigments which are helping in absorption of light from the mid part of visible light spectrum, widen the absorption of spectrum. So they protect chlorophyll from photooxidation. Similarly, the same thing happened in several types of cyanobacteria as well as other autotrophs. The absorption spectrum is different for different plants with different wavelengths of light observed by photosynthetic pigments. Whereas chlorophyll A and B absorb the maximum light in blue, violet, and red wavelength. Now, action spectrum shows maximum photosynthesis in blue, violet, and red part of visible spectrum. In the graphical representation shows the absorption spectrum between the photosynthetic system. Now, the absorption spectrum and the action spectrum coincide closely, showing that photosynthesis is maximum and blue-red regions, which was later found by Emerson in 1957 and named it as Emerson first effect and second effect. The first effect is also called as a red dwarf, as well as the second one is called as Emerson enhancement effect. Now, this is due to the presence of different types of harvesting molecules around the trap center in the photosynthetic unit. And of course, the presence of two interconnected pigment system, which is also called as photosystem 1 and photosystem 2, which are organized on the inner side of the thylakoid membrane into two discrete photosystems. So each photosystem has a specific chlorophyll A and many other pigments bound by proteins. The chlorophyll A forms the reaction center, whereas the actual reaction takes place while the other pigments, which is also called as light harvesting complex LHC, also called as antenna. Now, the photosystem 1, also known as reaction center P700, helps in the absorption of light at 700 nanometer. The electronic carriers involved in PS1 are pyridoxine, cytochrome complex, plastoquinone, cytochrome F, and plastocyanine. Now, the PS1 participates in cyclic photophosphorylation as well as non-cyclic. In the diagram representation, you can see how the photons are activated. Activates the photosystem 700 photosystem 1, and it releases the other proteins called pyridoxine, cytochrome complex, cytochrome F, and plastocyanine. So in the second system, that is photosystem 2, which has a reaction system at 680 nanometer, and the electronic carriers involved here are quinine, pheophytine, plastoquinone, cytochrome complex, plastocyanine, which is again common in both PS1 and PS2. So this happens with the splitting of water molecules, which releases the electrons for the activation of P80. And later on, the other proteins are formed with energy release, that is NADP. The light gathering or energy capture, it depends on light absorption and energy transfer, as you see in the graphical representation. So here the energized electron from the reaction center is accepted by the chemical acceptor, and the oxidized reaction center is reduced by electrons from inorganic or organic molecules. So here the water splitting mechanism helps the entire mechanism. The pigment antenna is a mechanism for gathering quanta in low light to drive a process which requires several quanta. So this helps the reaction center to gather maximum light from visible spectrum. The entire photosynthetic unit, assemblage of chlorophylls and other accessory photosynthetic pigments and reactions constitute the photosynthetic unit. The light harvesting system is made up of 250 to 400 pigment molecules, including PS1 and PS2. So here we'll end up the class, and the mechanism of photosynthesis will be discussed in the next class. Thank you.